ABTest service for the multifractional quantification of beta amyloid in plasma. More information for better science. Araclon Biotech is a company dedicated to the research and development of therapies and diagnostic methods for neurodegenerative diseases. Through investigation at Araclon, we have found that A-beta peptides can be found and measured in two fractions in plasma: the fraction free in plasma and the fraction found bound to plasma components. After a simple blood draw, and following the Araclon standardized sample protocol, we submerge in the processed plasma sample where we can see the fraction free in plasma for A-beta 40, A-beta 42 and A-beta 17, and the fraction bound to plasma components for A-beta 40, A-beta 42 and A-beta 17. ABTest service allows us to independently measure each peptide, A-beta 40, A-beta 42 and A-beta 17, in both the fraction free in plasma and the fraction bound to plasma components, with reproducibility inter- and intra-laboratory results empirically validated following clinical and laboratory institute guidelines to support a robust service. ABTest service. A validated ELISA colorimetric assay coupled with an automated system. Araclon manufactures all the chemical components that go into making up the A/B test. Araclon has in place exhaustive quality control protocols to ensure the high quality of its components, both individually and in combination. All this, plus a wealth of specialized experience in the quantification of A-beta in blood, coupled with investigation and new information generated on a continuous basis, allows us to deliver a robust service. ABTest service provides information on the total A-beta in plasma, the fraction free in plasma, and the fraction bound to plasma components. ABTest service can provide up to 6 direct results per sample, 2 fractions for each of the 3 peptides, and the ratios contained there within. Through past challenges, the investigation community has learnt many valuable lessons. One of these being that to answer the key questions from clinical trials, the right participants must be enrolled. Increasingly, this is accomplished with the help of biomarkers. Currently, to enroll 200 cognitively normal amyloid PET positive individuals between the ages of 65 and 70, one would need to screen 830 individuals using amyloid PET scans alone. These data are results from a collaboration between Araclon and the ABLE group. We have found that our ratio, total in plasma 42/40, may allow us to help in the enrichment of individuals for recruitment into clinical trials and other studies. With 72% sensitivity with relation to amyloid SUVR as measured by amyloid PET scan, combined with the natural prevalence, we would need to select a group of 1,153 cognitively normal individuals in the target age group. With the aid of the ABTest service in the recruitment step, we would select 282 individuals from the initial 1,153 deemed ABTest positive. ABTest could reduce the number of PET scans for screening in this scenario by 548. A confirmatory PET scan in those 282 individuals would eliminate the remaining PET negatives from the study population, substantially reducing the costs in the recruitment stage. To summarise, with the utilisation of ABTest to recruit 200 PET positive cognitively normal individuals, one could reduce the number of PETs performed in the recruitment step by 548 scans. An uncomplicated service. ABTest service uses a blood sample, easy, minimally invasive extraction. A simple sample protocol, logistics system for the collection, storage, transportation and delivery of samples. Same validated ABTest service in both Araclon laboratories. We are ready to offer the ABTest service for the reliable quantification of A-beta, using a validated assay, sample collection, storage and logistics protocol. ABTest service. More information for better science. This is our reality. What's yours?